Alright, so the focus of this video is on nervous system communication within and between neurons. So a big part of human stimuli is the actual, before we can respond, is we need to actually receive the communication and we have to receive the stimulus. So this is going to take you through what's happening at a cellular level in the nervous system. So before we get into looking at the cell membrane of a neuron, we need to think about what is stimuli. So stimuli is something that can be perceived by the senses and that your body can ultimately respond to. So um, that can be changes in temperature, it can be pressure or pain. Um, that can be picked up through mechanoreceptors, but there's also mechanoreceptors that are sensitive to vibrations in your ear. You have different receptors in your eyes that pick up variations in light, photoreceptors. We also have chemoreceptors inside of our body, but also picking up chemicals from outside the environment, like odor molecules. From there, through our nervous system, we can pick up these odor molecules and send electrical messages up to our nerves and ultimately to our brain to uh, have an idea or actually integrate that information. So we're going to look at this, and this is a diagram of a neuron. We have dendrites, which receive communication, axons, which send communication, and we have synapses, which are connections between neurons. And we're going to talk about how we get from receiving the stimuli and sending it through the neuron, but also how do we jump this little gap with neurotransmitters from one neuron to the next and continue the communication on. So we are going to focus first on communication through a neuron, and we're going to look at a cross-section of the cell membrane. So it first helps to understand a little bit about the electricity involved and some of the ions that we're dealing with here. So when we start talking about voltage, we're talking about a potential difference between electrical charges, and the current is the flow of a charge, movement of a charge. So here we first have um, an overall negative charge intracellularly because primarily because of these protein anions, which keep things fairly negative on the inside. We also have a high amount of sodium ions on the outside. So we have a lot of sodium out here, but not very many protein anions out here. So the initial state is going to be negative on the inside with a lot of sodium on the outside and quite a bit of potassium positive ions on the inside, but overall it's going to be negative because of all these proteins which don't flow in and out. So if we're looking at this as the cell membrane, we have lots of channels and pumps in here. Outside the cell, we have lots of sodium ions. Inside, we have potassium ions. We have a lot of protein anions. So the resting potential here, right here, is -70. And at this point, it's not communicating. It's waiting for some type of a stimulus in order to communicate. That might be odor molecules. So the initial reception of a stimuli would cause the first sodium ion channel to open, which would cause sodium to rush in. And it would change this, we call it depolarization, would change the charge from a -70 to a +40. This +40 voltage would then trigger the next um, sodium ion channel to open because this is voltage gated. The gate on the channel opens due to a change in the voltage. And now that +40 is being propagated. We send the action potential message along through the inside of this membrane because we are triggering through voltage, through changes in voltage, for the continuing opening of sodium ion channels. And obviously this happens very quickly. And boom, we shoot the impulse along the inside of the membrane, along the entire axon, through the flooding in of sodium ions, changing the inner part to positive 40. Now, communication pulses. It's not just on all the time. So that positive 40 voltage needs to get set back to negative 70. How does it do that? It doesn't initially send out the sodium ions. It repolarizes by sending out the potassium ions. The positive potassiums go out. So then all of these are again voltage gated. So when we have this change in voltage, the potassium ions go out, which trigger the next potassium channel to open, and we repolarize this. And now these are on the opposite sides, but now we would be back to a negative 70. Okay, so it's pulsing. The message got sent because we propagated the positive 40, but then we reset the resting potential by sending out potassium ions and moving it back to um, negative 70. But in order to be ready for the next pulse, we've got to put the sodium and potassium ions back to where they belong. So we need to reset the resting sides of the membrane. So we reset the voltage, but now things are on the wrong side. So we need to reset the ion concentrations. So now we get involved with the sodium potassium pump. And the sodium potassium pump is going to move these things back where they belong. And that requires a lot of ATP in order to pump them back where they belong. And that's why your nervous system uses so much ATP and so much energy. Even though your brain is only 2% of your body mass, it uses 25% of your whole ATP output. So the sodium potassium pump is going to reset the ion concentrations. We're still at negative 70 here, but now we're moving things back where they belong with the sodium on the outside and the potassium on the inside. Now, how do we communicate between neurons? Because we just sent the message all along this axon, but now we have a gap between these. We have a synaptic cleft, and we have to jump from one to the next. So again, if we look at this model, we depolarize this area. We had all of these voltage-gated sodium channels that are sending this positive 40 voltage, propagating that towards the synaptic terminal. And at that axon terminal, we then trigger this one. Now, it's green, it's not red. So what is this one? It's not a sodium channel, it's a calcium ion channel. But again, it's voltage gated. So this opens when the positive 40 reaches, the calcium floods in, and that calcium can release these vesicles of neurotransmitters. It's transmitting between the two neurons. So we send the neurotransmitter out, and that is allowed to jump the cleft, jump the gap. So this neurotransmitter is then going to land on the receptor on the dendrite of the next neuron. That, you see the hiding channel behind this? This is a sodium ion channel, but it's ligand gated. So right now, there is no voltage to make this open. But we have a ligand or a neurotransmitter, which is going to cause the opening of the channel. That opening of that channel allows some sodium to flood in, which now creates the positive 40 action potential, which that voltage can trigger the next ones to open. And now we go back to what we were just looking at previously, where the voltage gated ion channels are being triggered to open and flood and propagate along the next axon. So that's how we get a message sent, because the odor molecules don't go up into our brain. The odor molecules stay in the roof of our nose, but the message gets sent through our brain through the flooding of sodium and potassium in and out of neurons, out of nerve cells. Now, to talk about this again, we focus on communicating through a neuron, then we also focus on communicating between neurons. And that has to get sent over and over and over and over, because it's not just like one neuron that communicates up into your brain, but then that message ultimately gets sent to the part of the brain that makes sense of the message. So we have our sensory cortex, which is in charge of interpreting or bringing in the messages, and then it can speak to the motor cortex to send messages back out efferently to cause a response. So again, on this cortex, on this outside of your brain, we've mapped all the different parts of your cortex here where messages get sent to perceived stimuli. But again, the same process of communication with propagating action potentials is also going to happen in motor neurons, sending messages away and back to the muscles of your body. Now, we often see things like this. Again, we have axons and axon terminals in the direction that action potential is moving. We see this depolarization when the positives come in, repolarizing when it goes back to negative, and then we have this transition back to a resting potential. So again, the resting potential is at -70. The action potential pushes it to +40, and then we have to reset the resting potential back to -70, and we also need to reset the ion concentrations now through the sodium potassium pump to get the sodiums on the correct side and the potassium on the correct side. So this one is basically showing the same thing, it's just a little bit more accurate because depolarization, repolarization, and resetting the resting potential are all happening basically at the same time, but on different parts of the membrane. So we depolarize one part, push it to +40 through the sodium coming in, which triggers the next voltage gated channel to open, which triggers. Then we also trigger the opening to flood um, the potassium out, which is repolarizing or bringing the voltage back down to -70. But as this process is going, we are triggering the next voltage gated channel to open, which causes potassium to flood in. But as that's happening and moving further down the axon, we are repolarizing it behind. So then the potassium gets flooded back out to push this part to a -70. And again, all of this is communicating through pulsing. And as this is going here, we are also using ATP in the sodium potassium pump to reset the ion concentration back to resting. So we depolarize, we repolarize, and we rest. We depolarize, repolarize, and rest. And that's where that graph comes in, where you have that check mark of an action potential happening. So this is a little bit more scientifically accurate as far as looking at this depolarization, repolarization, and resting, what's happening at each of those stages. And this would be depolarization, this would be repolarization, and then as we go back to the resting potential, we need the sodium potassium pump to reset the ion concentration, put the sodiums and potassiums back where they belong. So that's how the nervous system works, communicating along a neuron and between neurons. So hopefully that's helpful.